So welcome and thank you for joining us. My name is Dean Lee and I'm Professor of Physics at the Facility for Rare Isotope Beams, or FRIB. Today's public talk is part of an initiative at FRIB called the Advanced Studies Gateway. The goal of the Advanced Studies Gateway is to inspire people. We bring together researchers, innovators, creative thinkers, artists, and performers from all fields and strengthen ties between Michigan State University and the community. Today, we have the pleasure of presenting a public lecture by Professor Doris Tsao entitled Representing the Visual World. Professor Tsao is a professor of biology at the University of California, Berkeley, and an investigator at the Howard Hughes Medical Institute. She joined UC Berkeley in 2021, and prior to that was a professor at the California Institute of Technology. She studied biology and mathematics at Caltech as an undergraduate and received her PhD in neuroscience from Harvard University in 2002. Her central interest is in understanding neural mechanisms underlying vision. Her lab seeks to understand how visual objects and actions are represented in the brain and how these representations are used to guide behavior. Her lab is investigating mechanisms at multiple stages in the visual hierarchy, from early processes for segmenting visual input into discrete objects to mid and high-level perceptual processes for assigning meaningful identity to specific objects, to processes by which these perceptual representations are integrated into coherent scene representations. Techniques used include electrophysiology, functional magnetic resonance imaging, electrical microstimulation, optogenetics, anatomical tracing, psychophysics, and mathematical modeling. Tsao has received multiple honors, including the Sofia Kovalevskaya Award in 2004, Eppendorf and Science International Prize in Neurobiology in 2006, a MacArthur Fellowship in 2018, and a Kavli Prize in Neuroscience in 2024. She was also elected to the National Academy of Science in 2020. If you have any questions during this talk, please type them into the chat or question and answer dialog box. At this time, it is my great pleasure to introduce Professor Tsao.
Thank you so much, Dean. I'm delighted to be here. So I was so happy to get this invitation from Dean because he was my TA in Sidney Coleman's quantum field theory class back when I was in grad school at Harvard. And I have to say that taking and surviving that class was undoubtedly one of the intellectual peaks of my life. So I want to start with this joke that Sidney Coleman made in the class, and I love this so much, especially since I imagine some of you in the audience may be physicists like Dean. So, by a wonderful fluke peculiar to living in 3+1 dimensions, the representations of SO3-1 can be rapidly obtained from the representations of SO3. You know all about the representations of SO3 from undergraduate quantum mechanics, so we'll be able to wrap the catalog up by the end of next lecture. If we lived in 9+1 dimensions, there would be no quick reduction of the problem of finding the representations of SO9-1 to the problem of finding the representations of SO9, which 9+1 dimensional students solve as undergraduates. And I love this joke so much because it spoke directly to a central question I've been interested since I was in grad school, of how the brain represents the visual world. How can electrical activity in a sheet of microscopic neurons evoke the percept effortlessly, for example, of a mountain standing tall in 3D space? So neurons in the brain are obviously not building the mountain in the same way, say, that cells in the liver are building the liver. Instead, they're doing something more ethereal, creating a representation of the world from sensory energy. And the essential problem I want to understand is how billions of tiny neurons, each with limited computing power, can work together to solve the gargantuan task of representing the visual world. And I want to begin by discussing the general concept of representation. It's a tricky concept, though I think it's also what makes neuroscience special. So a physicist trying to understand why a certain material has certain properties never has to worry about what the material is representing. She just has to build a model of material that makes correct predictions. But neuroscience is all about representation. There's a subjective mental world of percepts, thoughts, and emotions, and this is the mind. To us as neuroscientists, it seems obvious that everything that happens in the mind depends on or is something that happens in the brain, which exists in the physical world. And we want to develop a comprehensive scientific explanation of all mental events. But of course, these mental events are trapped in a subjective world that we each experience only through a private consciousness. At the moment, it's difficult to imagine how any scientific theory could explain how neurons in the physical world can generate a private subjective world. But I believe that everything we experience is precisely describable. And as I hope to show you, by studying neural representations, we can find a language to make previously inexpressible experiences explicit and concrete. And the hope is that by reducing this gap, we may be able to see to the other side. Now let's move away from the mind-body problem to talk about the actual brain and how it sees, because the brain evolved not to ponder philosophy, but to eat. As primates, our visual object perception abilities are extremely impressive. We can effortlessly recognize a seemingly infinite number of different objects. And we can do this even when they vary in subtle ways. Moreover, we can recognize the same type of object even when its appearance changes drastically. So the brain's representation of the world is very different from the raw sensory input. Indeed, even the exact same image can evoke different subjective percepts. And today I want to show you how we're trying to understand this problem of representing the visual world. And I want to emphasize at the outset that we're still at very early stages, and many questions, for example, the mystery highlight in the slide of how the same sensory input can lead to different percepts, remain unknown. And so my talk today, I first want to tell you about the brain organization and code for faces, which is a problem my lab has studied for many years. And then I want to tell you about the organization code for objects in general. And then I want to revisit the code for faces and tell you about some recent surprising insights into the neural dynamics of representation. And finally, I want to talk about the neural code for face memories. Okay, so let's get started. The macaque face patch system is an amazing gift from Mother Nature for systems neuroscience. So here's a side view of the macaque brain, and decades of experiments have revealed a hierarchy of areas that represent different aspects of the visual world, starting from local edge orientation in V1, and then proceeding to more complex features in higher regions. In particular, cells in a region called infratemporal, or IT cortex, represent the visual form of high-level objects. So lesion in this part of the brain can lead to selective inability to recognize specific visual forms such as faces. And back when I was a grad student, using fMRI in monkeys, or functional magnetic resonance imaging, I found 6 patches of cortex in IT, in this large region of infratemporal cortex, that respond highly selectively to faces. And they're shown here in yellow. And each of these patches turns out to contain an extremely high concentration of face-selective cells. So what do I mean by face-selective cell? Now I want to show you a movie of one neuron from the middle face patch. Okay. And so this is one of the first neurons I ever recorded back in 2004, it's 20 years ago now. And so you can hear what this cell sounded like. And so the clicks represent the action potentials, or the electrical pulses fired by the single neuron. And you're going to see the same sequence of images that the monkey saw when we recorded from this neuron. Okay, so I hope you can hear how selectively this cell responded to the faces. So that's an example of a face-selective neuron. Zooming back out to the system as a whole, the 6 patches are strongly and specifically connected to each other, forming an interacting system that gives us a whole sequence of nodes to target, to probe how representation of a face is successively built up in IT cortex. Indeed, we found that each face patch performs a different step in face processing. In particular, cells are increasingly view invariant as you go anterior. So in the middle face patch, single cells respond to faces at a specific view, like this neuron, which prefers left profiles, while in the most anterior patch, cells respond across all views, like this neuron. And overall, the space patch network provides us with a remarkable system for studying the neural code underlying high-level object representation. And in particular, the extraordinary concentration of face cells within the face patches presents an exciting opportunity to study in detail the feature code used by the brain to represent this ineffable thing called facial identity. So can we leverage our access to face cells to understand how facial identity is represented? And this question was tackled by a brilliant postdoc in my lab, Stephen Le Chang, who now heads his own lab at the Shanghai Institute of Neuroscience. So let me begin by defining feature code more precisely. The feature code is the mapping or dictionary between the stimulus and responses of neurons. And if we understand this mapping, then we should be able to predict responses of cells to an arbitrary stimulus. And if the mapping is faithful, then we should even be able to reconstruct the stimulus from the responses of cells. And if both of these were possible, then it would really show that we know the code, the language the neurons are using to describe faces. So to figure out the brain's feature code, we first needed a way to parameterize faces. I describe every face by a set of numbers. And we considered various computer vision models for doing this, and I'm going to tell you what we found with one of the models called the shape appearance model. So this model describes every face by a set of 50 numbers. And the first 25 numbers describe the shape of the face. Right? So here's a face undergoing changes in these shape parameters, and you can see they capture the location of key landmarks on the face. The second 25 numbers describe the so-called appearance of the face. So here's a movie of a face undergoing changes only in the appearance parameters, and they capture the texture map of the face, like the complexion and so on. And so here you can see on the right, the landmarks are fixed, but the texture map is changing. And so this shape appearance model creates a 50-dimensional space of faces where every point is a face. And now that we have a way of parameterizing faces, we can generate a stimulus set to probe how the brain represents facial identity. And in our stimulus set, we randomly drew 2,000 faces from the 50D space and presented them to the monkey while recording cells from 2 face patches. And importantly, we showed the same 2,000 faces for every cell, so we could systematically understand how each face is encoded by a population of neurons. And as a starting point, we can plot the tuning of cells to each of the 50 shape and appearance parameters. And so here you can see tuning curves of 2 example cells to the first 3 shape parameters and the first 3 appearance parameters. And cell 1 was tuned to this shape parameter, and cell 2 to these 2 appearance parameters. And you'll notice that all 3 curves here are ramp shaped with a minimum at one extreme and a maximum at the opposite extreme. And this was true across the population. And these ramp shaped tuning functions mean that each cell is acting like a ruler, measuring a face along a subset of dimensions. And mathematically, if a face cell has ramp-shaped tuning to different features, this suggests that its response can be roughly approximated by a linear combination of face features, where these coefficients are simply the slopes of these ramp-shaped tuning functions. And this has an extremely powerful consequence. Namely, for a population of cells, we can write the population response vector as a linear transform of the 50 face features. And then we can simply invert this equation to predict the 50 face features from the population response vector by simple linear transform. In other words, we should be able to reconstruct faces from neural activity by simple linear transform. So is this true? We next trained a decoder to do exactly this. We used responses to all but one of the 2,000 faces that we showed to the monkey to learn this weight matrix. And then we used the responses to the left out face to reconstruct the 50 face features using this learned weight matrix. So how good is this reconstruction? Here's an actual face that was presented to the monkey, and here's the prediction from linear regression using activity of just 205 face cells. And here are 3 more examples. And you can see the reconstructed faces match the actual faces almost exactly. So I like to say a picture is worth 1,000 words or 200 neurons, plus $1 million of NIH funding. So we can reconstruct the stimulus from neural responses. And moreover, from data I haven't shown you, it turns out that we can also predict responses using the shape appearance model, explaining around 80% of the explainable variance of face cells to these faces. And our ability to go both forwards and backwards suggests that we really know the brain's code for this ineffable thing called facial identity now. And a simple geometric interpretation of the feature code used by a face cell is that cells are projecting incoming faces onto a specific preferred axis, because dot product is projection. And this geometric picture implies that a face cell should respond exactly the same to all faces in the plane orthogonal to the preferred axis, right? Because all these points should have the same value of projection. And this highly surprising prediction hands us a strong test of whether our understanding is correct, and the code used by face cells really is an axis code. And in fact, the prediction turned out to be true. So here are responses of a face cell to the 2,000 faces projected onto the cell's preferred axis on the x-axis and an orthogonal axis on the y-axis. And each dot represents a response to one face, with red indicating high firing and blue indicating no firing. These 3 red dots represent responses to these 3 identities. These 3 different faces look like very different people, but they all elicit the same strong response in this one cell because they each have the same value of projection onto the preferred axis. And the difference between them lies along the dimension orthogonal to the preferred axis. And without understanding the axis code for facial identity, it would have been entirely non-obvious what linked these 3 identities. So why does the brain represent faces by a shape appearance axis code? I think there's actually a beautiful explanation, and it comes from a powerful idea about what visual perception is trying to achieve, called the analysis by synthesis hypothesis. And the idea here is that the central goal of visual perception is to create a generative model of reality that is as parsimonious as possible. And let me try to unpack this. So you can think of a generative model as something like the knobs of a video game engine that lets you create any scene you want with any number of characters, objects, and so on, once you know the value of each knob. And some of you, I'm sure, have played with DALL-E or Stable Diffusion, and these are generative AI models that let you create images just from text captions. And a remarkable thing is that the size of the Stable Diffusion model, which lets you create a 512 by 512 image of virtually anything you can possibly imagine, is just 4.2 gigabytes. This is vastly smaller than the size of all possible 512 by 512 images. So Stable Diffusion is taking a text prompt and generating an image. And the idea of the analysis by synthesis hypothesis for neural representation is that the brain is doing the reverse. It's taking the incoming image and extracting a representation that's hugely compressed. So let's see, yet it's still rich enough to reproduce the input exactly. Okay, so here it would certainly need to be slightly richer description than just cute, magical, flying dog. So the key insight here is that the world of physical reality is much lower dimensional than the space of all possible images. And we just want the brain to accurately and explicitly model physical reality. And I think this explains why face patches are using a shape appearance axis code, because this code is precisely a highly parsimonious generative model for faces that represents any face by just 50 numbers sufficient to regenerate the face. And indeed, if we compare the shape appearance model to different models for parameterizing faces that is not a generative model, such as VGG face, which is a deep network trained to simply recognize different faces, we find that the shape appearance model does a much better job at explaining macaque face patch activity, because the code used by face patches is not optimized only for face recognition, but it enables reconstruction of the face useful for any task. So to summarize the functional anatomy and code for faces in IT cortex, IT contains a network of connected patches. Cells within the network show consistent selectivity for faces, and there's increasing view invariance along the network. And with respect to the neural code, cells throughout the face patch system represent faces using a shape appearance axis code. And this is a highly parsimonious generative model for faces that's predicted by the analysis by synthesis hypothesis. So that's how we think IT face patches code faces. Now let's turn to the question of how the brain represents general objects. And this work was led by an amazing postdoc, Pinglei Bao, who now heads his own lab at Peking University. So can what we've learned about faces help us to understand the rest of IT cortex? And this is a much more challenging problem because IT cortex is huge, and the parameter space involved in representing an arbitrary object is also huge. Besides face patches, specialized networks have been found in IT cortex for bodies, scenes, and colored objects. But there's still large parts of IT that are completely uncharted and don't show a differential response to any known stimulus comparison. Are there any principles governing the organization of this unmapped territory? So before we could venture into this unmapped territory, we first needed to identify it. So we began by mapping all of these known regions in a monkey. So then we could tackle these unknown regions. And we started with an exploratory approach by electrically microstimulating a piece of this unmapped territory while performing simultaneous fMRI to ask, what other parts of the brain is this piece of cortex connected to? Okay, so again, this piece of cortex that we're going to electrically stimulate while scanning the monkey is not activated by any known localizer. It's not in a face patch or a body patch or a scene patch or any known patch. Okay. And when we stimulated in this region, here you can see the activation pattern when we compare stimulation versus no stimulation. And so not surprisingly, we saw some fMRI activation at the stimulation site. But in addition, we saw activation in these two discrete regions here, one more posterior and one more anterior. And here you can see the fMRI response time courses from this posterior site and from this anterior site. Okay, so every block here represents the presence of electrical microstimulation. And so what this means is that these two regions are connected to this region. And so we've discovered a new anatomical network of connected patches in this unmapped territory of IT cortex. So we're going to call this network X for the time being. And to begin understanding the function of network X, we presented a stimulus set consisting of 51 random objects, each at 24 different views. And here you can see responses of cells from the 3 patches of network X to these 51 objects. So each row is one cell, each column is one object, and the cells are grouped separately for the 3 patches of network X. What you see is that there's very high consistency in the selectivity of cells within this network. All the cells in X1, X2, and X3 are suppressed by faces, and many show a strong response to a few of the objects shown here. And you'll notice these objects are distributed across the different categories. They all seem to us to have in common that they have these protrusions. Okay, so we found a new functional network in IT whose cells show consistent selectivity, but the network is not specialized for a specific category the way the face patch network is. So as I mentioned earlier, one of the hallmarks of the face patch network is this increasing view invariance as you go anterior. And we also looked at the view invariance of cells in different patches of network X, and we found that it was strikingly similar to the face patches. So cells in the most posterior patch of network X were view specific, while those in the most anterior patch were view invariant. So we see these intriguing similarities between network X and the face patch network. And this led us to ask, do cells in network X represent object identity using an understandable code, as we had found for the face patches? And the key challenge was to develop a way to parameterize arbitrary objects. And until a decade ago, this was an impossible problem. But now, with deep networks trained on object classification, we have a great solution. So, as many of you know, deep networks were originally inspired by visual neuroscience, and now we can also, in turn, benefit from them. So for every object, we can pass it through a deep network to get out a few thousand numbers describing the object at a high level, which can be used to classify the object identity with the linear classifier. And then we can use principal components analysis to further reduce the dimensionality of this space to just 50 numbers. So now we have this 50-dimensional space of objects. What do the resulting axes look like? So here you can see objects with the highest and lowest projection onto the first PC. And these PCs were computed using AlexNet layer FC6. And here's the second PC. Okay. And so just for the sake of being able to refer to these axes, we call them by what they look like. So this is spiky versus stubby, and this is animate versus inanimate. Okay. So when we analyze the responses of cells in network X to objects parameterized by these object axes extracted from the deep network, we found that they behave very much like face cells, with single cells showing ramp-shaped tuning to subsets of object parameters. And this suggests that object cells are using axes to represent objects just like face cells. And so to summarize the functional anatomy and code of network X, it's strikingly similar to the face patch network. It consists of a network of connected patches. Cells within the network show consistent selectivity. There's increasing view invariance along the network, and cells represent object identity using an axis code. So there seems to be a clear structural parallel between the face patch network and network X, suggesting that IT may obey a more general principle of organization. So network X covers just a portion of this unmapped territory of IT. Given its remarkable similarity to the face patch network, this made us wonder whether there are other similarly organized networks in IT. And if so, is there a general principle governing how all of these networks are arranged? The fact that we found network X through random targeting initially suggests that either we were incredibly lucky or it must not be unique. So we decided to take a second look at the networks that we had found. And recall the first two axes of object space are roughly stubby versus spiky and animate versus inanimate. Faces are stubby, animate objects. Bodies are spiky, animate objects. Network X seemed to prefer spiky objects, regardless of animacy. These observations made us wonder whether it's possible that all of IT would actually be topographically organized according to these first two dimensions of object space, in the same way that retinotopic cortex is organized according to polar angle and eccentricity. And as a first step to test this, we projected each of the stimuli that we showed to the monkey onto the first two dimensions of object space. So each stimulus here is one of these gray circles. And then we marked the top 100 most effective stimuli for network X, the body network, and the face patch network. And you can see that they approximately span 3 quadrants of the space. But there's a region of this space that's not represented by any of the cells. And if it's true that IT cortex is laid out according to the general scheme shown here on the left, using the first two dimensions of object space, then we predicted that there should be a fourth network representing objects projecting strongly onto this region, namely stubby, inanimate objects, lacking protrusions, like a USB stick or a radio. And this was an entirely non-obvious prediction because we had actually, before making this prediction, never recorded from a single cell preferring such objects, let alone a whole network of such cells. Okay, we thought they were just boring objects. And to test this prediction, we first ran an fMRI experiment with 4 different blocks corresponding to the 4 quadrants of object space. And when we compared stubby versus other blocks, we actually found a network containing multiple patches selective for stubby objects, shown here on coronal and sagittal slices. And here you can see the electrode entering the anteriormost stubby patch. And here are responses of 2 cells that we recorded from this patch. And you can see that both of these cells strongly preferred stubby objects, like this truck and these houses. And here you can see the population plot from this stubby patch network, with the 5 most preferred and least preferred objects below. And you can see it satisfies our prediction, and this response pattern is very different from network X and also from the body patch network, and of course the face network. So the hypothesis that IT cortex is organized according to the first 2 dimensions of object space actually let us discover a new shape network. And to my knowledge, this remains one of the only cases so far where deep networks have actually let us discover a new phenomenon about the brain, rather than just confirming something that we already knew. And when we projected the preferred objects of the stubby patch cells onto object space, we found that indeed they mostly lie in this previously unrepresented region. And here you can see the 4 networks overlaid on a coronal slice in posterior IT. You see a progression from body to face to stubby to network X in both hemispheres. And the spatial organization is preserved in middle and anterior nodes of these networks: green, blue, purple, orange, green, blue, purple, orange. And it's also preserved across animals. And so to summarize overall, here's the current big picture of the spatial organization of IT cortex for representing object shape. IT contains at least 3 distinct mappings of object space, with increasing invariance from posterior to anterior. And each mapping is further split into at least 4 regions tiling the 4 quadrants of object space. And faces occupy one of these quadrants, but importantly, the other quadrants are represented using a similar architecture and coding principles as for faces. And using activity from just a few hundred cells recorded from these 4 networks, we can actually reconstruct arbitrary objects. And so here you can see some example reconstructions. And while they're certainly not perfect, they confirm the richness of the object code contained in just a few hundred neurons within these 4 networks, which is a tiny fraction of the roughly 50 million neurons contained in all of macaque IT cortex. So I think what's really exciting here is that we've changed the basic understanding of IT cortex. For the first time, we provide a unified model for the functional organization of IT cortex in terms of object space. So are we done? Have we solved the problem of visual object recognition? Far from it, and that's what I want to talk about next. So the basic paradigm of sensory systems neuroscience has been to show lots of different stimuli to an animal, measure the responses of neurons in an area of interest, and try to decipher the transformation between stimulus and spikes, which we call the neural code. And so far, my talk has squarely followed this paradigm, right? I've suggested to you that by showing all these faces and objects to the cells and face patches and the rest of IT cortex, we've gained a pretty good understanding of the neural code used by cells across IT cortex for representing high-level visual form. And it's something like the code used by a deep neural network trained to analyze objects. But it turns out this is actually not true. So the neural code used by a single neuron in IT cortex can actually change completely. A neuron can switch back and forth between totally different neural codes, but the switching occurs on very fast timescales, as I'm going to show you next, from on the order of tens of milliseconds. And so all the studies I showed you before have averaged across a long time window, and so they were blind to these changes in neural code. And the switches in neural code that I'm going to tell you about next that are happening on these timescales of tens to hundreds of milliseconds are interesting because they're not random, but we think they're serving an important function for visual processing. And to jump the gun slightly, I think the bigger message that these changes in neural code convey is that we need to revise the current paradigm of focusing on single areas. Just as we can't understand tides on Earth without knowing about the moon. So I believe the changes in neural code that I'm going to show you imply the need to understand interactions between areas. Okay, so now I want to tell you about this neural code switching for representing faces that we discovered recently. Okay, so the big picture I described, that faces are processed just like other objects, was satisfying, but it was also puzzling because it violates a claim that psychologists have made for many decades that faces are processed by specialized or quote-unquote domain-specific mechanisms. And this claim is based on demonstrations like the Thatcher illusion here, where you can tell apart these two faces much better when they're upright. And this led to the notion that when visual input enters the visual system, there's an initial stage where the brain decides if the image contains an upright face or not. And only if it passes this stage, then the image proceeds to be analyzed by a set of specialized mechanisms for measuring face features that only work on upright faces. But if all of IT cortex, including face patches, is something like a general purpose feed-forward deep neural network, then there's no such thing as domain specificity. Each unit in a deep network processes every image in exactly the same way. The units aren't doing something different for a picture of Marie Curie compared to, say, a picture of a tree. Instead, every image gets passed through the exact same set of filters. So we decided to examine this question: do face cells encode faces and non-face objects in the same way, like units in a deep network, more deeply? And this study was led by a very talented graduate student, Yuelin Shi, together with another graduate student, Dasheng Bi. To address whether face cells encode faces and non-face objects in the same way, we did a simple experiment. We first measured responses to a large set of faces, and we modeled the responses of each cell as a linear combination of 60 features of each face, which I already told you about. But importantly, each face is now going to be described by features in a general 60-dimensional deep neural network-derived object space. And this is important because we want to compare encoding of faces and objects in the same space. And so we call these 60 regression coefficients the face axis of the cell, as we've already discussed. And then we measure the responses of the same cells to a large set of non-face objects to get out a set of 60 numbers that model its response to features in non-face objects. So these 60 numbers define the object axis of the cell. And the key question is, are these two encoding models for faces and non-face objects the same? So within the 60D general object space, is the face axis mapped with faces the same as the object axis mapped with non-face objects? If so, this would suggest that face cells really are like units in a feed-forward deep neural network, and it would demolish the idea that face cells are somehow specialized for processing faces. So here you can see all the objects that we showed to the monkey projected onto the first two dimensions of the general object space. So here, each dot represents the position of one non-face object in the feature space. And these non-face objects all cluster in this lower left area. Here, these dots show the positions of the face stimuli in the same feature space, and they all cluster in the upper right quadrant. And they cluster separately because faces have distinct set of features. We can color all of the non-face objects based on the responses of this example cell to each of the images. And you can see this response gradient pointing to the upper right. In other words, this face cell responds more and more to non-face objects as they become more face-like, apples and buttons and so on. We calculate the cell's object axis, and the first two dimensions are shown here as the green arrow. So now what happens for faces? We can again color all the face dots based on the cell's response to these face images. And astonishingly, the cell's face axis and object axis turn out to be totally different. In fact, they're pointing close to opposite directions. So this cell uses a totally different encoding model for faces compared to objects. Here are the object and face axes for all the cells that we recorded. And you can see that the face and object axes are totally different. The object axes for all the cells consistently pointed to the upper right quadrant, which is where faces are located in the space. So this means that when you map the cell with non-face objects, the cell is wired to detect features diagnostic of a face, i.e. it's doing face detection. Whereas the face axes are much more scattered and better suited for telling apart differences between different faces, i.e. performing face identification. But how can a cell decide which axis to use when it first encounters an image? We wondered if cells might actually make this decision in a dynamic way. So for calculating the face and object axes, so far we've been using the responses averaged across a relatively long time window. We next decided to use a short 20 millisecond sliding window so we could see in detail how the encoding axis develops over time. Now I'm going to show you snapshots of the face and object axes for an example face cell at 2 different time points. So here you can see the face and object axes of this cell 80 to 100 milliseconds after stimulus onset. And this covers the initial response of the cell. And you can see they point in the same direction at this time point. And now, just 20 milliseconds later, something dramatic happens. The object axis stays pointed in the same direction, but the face axis reverses. And we found this initial alignment and subsequent reversal of the face axis in almost all the cells. And to quantify this change in neural code, we computed a so-called similarity matrix. So every point here represents the mean correlation between the object axis at one latency and the object axis at a different latency. And the solid block of orange shows that the cell's object axis stabilized very early, around 50 milliseconds. Here you can see the similar matrix for the face axes, and they stabilize later, around 120 milliseconds. And here was the astonishing finding. So if you look at the correlation between the face and object axes, you can see that the face axis is initially aligned with the object axis. But then it becomes negatively correlated with it starting around 120 milliseconds. And you can see this change happens quite suddenly, suggesting a concerted change in the neural code for faces across the population. So to recap, we found that a cell's face axis aligns with its object axis early on, meaning that the cells are using a general coding mechanism early on. And both the face and object axis at this early time point are pointed towards the face quadrant, meaning the cell is optimized to detect faces. But then the face axis diverges from this general axis, and this divergence only occurs for faces. And this diverse tuning enables cells to discriminate between different kinds of faces. So face cells achieve specialization over time through a rapid concerted change in the neural code. And this change in code shows that IT cortex is not just a feed-forward deep neural network. And from additional analyses I don't have time to show you, we know that this code change is not due to adaptation. It involves changes in many features. The low-dimensional features become anti-correlated, so you're basically subtracting at long latency everything that faces have in common, right? While the high-dimensional features become uncorrelated from what the tuning at early latency. And this code change has a measurable functional consequence, leading to better face identity decoding at long latency. Okay, so just to summarize again, at short latencies, face cells use a general purpose code for faces and objects optimized for face detection, while at long latency, face cells use a specialized code optimized for face discrimination. Now in the last part of my talk, I want to switch gears and tell you about the neural code for face memories. And here again, we're going to see a major code switch happening at long latency, but now for a different purpose related to memory. And this was led by a very talented former postdoc Liang Shi, in collaboration with Marcus Benna and Stefano Fusi at Columbia. So how does the brain actually recognize a face? This involves first extracting the features of an incoming face, but then a further step is required to compare the incoming face representation to a representation in memory. And a long-standing assumption of neuroscience is that the same neurons that encode sensory stimuli also are responsible for storing long-term memories of those stimuli. And this suggests that these IT cells, which carry this continuous axis-based coding scheme, should somehow also support object memory. But schemes for representing memories often invoke attractors that would lead to breakdowns in continuous representation. And this was, the subject of the Nobel Prize this year to John Hopfield in physics. So how can we reconcile this discrepancy in computational requirements for efficient feature representation on the one hand versus memory on the other hand, if the same neurons are doing both? So we decided to tackle this by examining in detail how familiar faces are encoded in the face patches. And we began by presenting a set of screening stimuli consisting of human faces, monkey faces, and non-face objects, and these were either personally familiar or unfamiliar. And the unfamiliar images were shown just once per cell. And we confirmed behaviorally that the monkeys actually perceived the familiar and unfamiliar faces differently with a preferential looking test. So humans like to look at novel things, and so did our monkeys. They showed greatly increased viewing time for the unfamiliar faces. Now here you can see responses of one example cell from the most anterior face patch to our screening stimuli, and the cell responds strongly to both the familiar and the unfamiliar monkey faces. And to fully characterize the response selectivity of the cell, we measured responses to 1,000 parameterized monkey faces, which were all unfamiliar. And here you can see the responses projected onto the cell's preferred axis on the x-axis and orthogonal axis on the y-axis. Each dot is a response to one unfamiliar monkey face, and red again is high firing, blue is no firing. And you can see this very clear ramp-shaped tuning along the preferred axis, exactly as expected for a cell with axis tuning to face features. If we project the responses to the unfamiliar faces onto this axis, we see that they closely follow this ramp. Now here you can see responses to the 8 familiar faces projected onto the same axis. And now you can see they don't obey this ramp at all. Right? And this was typical of the population, and we quantified this as follows. So for each cell, we computed the preferred axis of the cell using responses to the large set of unfamiliar faces. We correlated this to the preferred axis using a left out set of unfamiliar faces. And here you can see the distribution correlation coefficients. And then we repeated this procedure for a set of familiar faces. And here you can see the correlation coefficients. Okay, so this shows that familiarity is causing a code switch. And I want to add that Liang went to extraordinary lengths to match the feature distributions of the familiar and unfamiliar faces to make sure that this change in encoding axis was not due to some kind of stimulus difference, but it could really be attributed to memory. So can you tell which of these two sets of faces is familiar and which is unfamiliar? Probably not, because you didn't know any of these monkeys, but the face cells in the monkey that we were recording from definitely could. And so next we repeated the previous analysis in a time-resolved way, and we found that this change in preferred axis for familiar faces occurred at long latency, well after the initial response. And here you can see a reconstruction analysis that visually confirms this representation change. So here are two actual faces that we showed to the monkey, one familiar, one unfamiliar. And here's the reconstruction using the short latency response together with the decoding model that was trained on a large set of unfamiliar faces. And you can see that both of these reconstructions are reasonable because the code for familiar and unfamiliar faces is the same at this time point. Here's the reconstruction using the long latency response. And now the reconstruction for the familiar face is totally off because the brain is using a different code to represent familiar faces at long latency. Okay, so at long latency, face cells are using a different neural code to represent familiar faces. So an obvious question is, why do face cells use a different code to represent familiar faces? So axis coding implies that the response of each face cell is a linear transform of this low-dimensional face feature vector. And geometrically, this implies that responses to faces are confined to this low-dimensional linear subspace of the full neural population space. So if you have, say, 1 million neurons in a face patch, this represents a tiny slice of the full response space, just 50 out of 1 million dimensions if you're describing faces by 50 dimensions. Okay. By pushing the response to familiar face off of this subspace, one can then easily separate this face from all other faces, creating a lot more coding space to build attractors around specific identities. And so our conclusion is that memory unfolds over time each time you see a familiar face. The response to a familiar face initially lands on the 50D subspace, but then it gets pushed off, while responses to unfamiliar faces land on the 50D subspace and they stay there. And my hunch is that this is a major reason why we have so much cortex. Even though we can reconstruct a face with just 50 neurons, I suspect all of that cortex is there for storing memories and context. And so I've shown you these two examples where the neural code rapidly switches in IT cortex for representing face identity and face memory. What does this code switch mean? So one big lesson I take away is that the brain really can't be explained by feed-forward chain of processing from input to behavior. Because otherwise, the degree of specialization that would need to happen in downstream areas so that they always use the right decoder to interpret IT activity correctly across all these switches in neural code would just be unwieldy. Instead, the brain is a dynamic system of interacting nodes, and we can imagine that high-level nodes are continually interrogating the low-level nodes with different questions, and the neural code that's used depends on the question. So what is present? Who is present? Do you remember this person? Etc. And to get at these interactions between areas, we're now going full bore into showing monkeys movies of dynamic natural scenes and recording with these high channel count neuropixels probes all over the brain in many areas beyond IT cortex. And one surprising finding so far is the extent to which every single area we've recorded has a surprisingly detailed visual representation. So hopefully, understanding the basic visual codes used within each of these areas will give us insight into how these areas are interacting. And I think that understanding these interactions between these areas is the next frontier of visual neuroscience. And so I hope you can see how by recording from single neurons across the brain, we can make progress into this gargantuan challenge of understanding how the brain represents the visual world. Thank you very much.
Thank
And
you
I'm
so much.
happy to take questions.
Thank you so much, Doris. That was an incredibly interesting talk. Really well put together, really exciting stuff. So if you have questions, go ahead and write them into the question answer dialog box. There are a couple of questions and I'll read them now. A question from Joe Nadowitz is, would you expect to find different response patterns if you were to perform same studies on other animals and species with different size brains?
Wonderful question. So I actually took a long detour into studying several other species, mice and tree shrews. I can say in mice, no one has found anything comparable to monkey IT cortex. And it's not, so you find tuning to orientation, the same way as you find tuning to edge orientation in monkey primary visual cortex. But then beyond that, this tuning, this very precise tuning to high level objects has been hard to come by in the mouse. And so that's why I switched gears to study the tree shrew, which is this highly visual mammal. It's one of the closely related mammals to the primate lineage. It's native to Southeast Asia, but it's still, it's a very small animal, right? And has very good vision because it lives in the forests. And what we found in the tree shrew was super interesting. So we did the exact same experiments as I've shown you here in the monkey, and what we found was that there was very compressed hierarchy, and area V2, which is just one synapse after V1, already in the tree shrew accomplishes many of the functions of monkey IT cortex. So it has cells selective for these object axes, and you can actually reconstruct complex objects using just a few hundred cells from the tree shrew area V2. So it seems like evolution led to this deeper hierarchy somehow. And of course, there's going to be more functions that we have yet to uncover, but all of that is actually present in tree shrew V2.
Very interesting. A question from anonymous attendee asking, I wonder what your results would turn out if toy faces, I think he or she is talking about like a doll or something that looks like a face, but not is a face, were used in your analysis. How would toy faces be distinguished from objects?
Yeah, so we actually, very early on, we showed a large set of cartoon faces that were parameterized by different parameters like inter-eye distance and eye size, so there were and we found that these the cells showed very strong tuning to these cartoon faces. So they're processing these toy faces in the same way. And I think that explains why cartoons are so effective. Yeah.
Yeah. Okay, there's a question from Yanzhou Ma. He asks, do you think these patches or building blocks of images in the brain were born as a default or learned at early ages?
Yeah, my PhD advisor, Marge Livingstone, has done many experiments to get at exactly this question. So she's raised monkeys without any face experience. And so and what she finds is that they don't develop face patches. So you really need face experience to get these face patches. That said, I think that they should have a network of patches for round things like apples and clocks, right? That was the implication from the first part of my talk, where I talked about how IT cortex organizes general object space. And so I think even without face experience, you should have that quadrant. But then with face experience, that piece becomes specialized for faces.
Okay. And there's a question from Tahir Saif, asks, what is the speciality of image cells? Will optogenetic neurons in vitro store and reconstruct images?
Mm. Yeah, I'm collaborating with my colleague Hillel Adesnik now to try to, generate artificial face percepts, right? So I showed you that we can reconstruct a face using activity of just 200 neurons. Of course, there's going to be millions of other neurons that are correlated to those 200 neurons, but it's a totally an open question. If we stimulate those 200 neurons in that pattern, which we can do now, technologically, um, will that evoke the face percept? Will that, stimulate these attractor networks, and so these other neurons will also get activated via the recurrent connections? Open question, but very interesting question with huge implications, I think.
Okay. Maybe I'll throw in one question. So it's really fascinating. I was curious why the linearity shows up so cleanly. Why wasn't there an activation function and you had to do some sort of an extra layer to find these correlations? Why is the linearity just showing up so easily?
Yeah, I think that the linearity is it's basically another way to say that we found the correct parameterization, right? If we have parameterized faces, say using the pixel value of every pixel, the contrast of every pixel, then it would be highly nonlinear, right?
Yeah.
And so I think your question, a different way to state your question is, why is the shape appearance model the parameterization that the cells are using? And I think there's a very deep reason for that, which is why the computer vision people developed this parameterization in the first place, right? The reason why they developed this parameterization was to create a model to track faces, right? So when you track a face, you want to be able to separate, decompose the representation of the identity that's indicated by the texture map from the changeable features, right? Which is these landmarks, so the shape parameters. So that's why they built this model, to separate shape and appearance features, because then that abstracts the appearance features from the shape parameters. And I think that's exactly why the brain is using this parameterization, because it also wants to be able to track identity invariant to head orientation and other changes.
Okay, we'll take one last question. John Allen raised his hand. Let me just allow him to talk. John, why don't you just ask your question? Go ahead. Go ahead, John. You,
Maybe you're muted.
yeah.
Oh, no, you're not. Yeah, we can't hear you.
All right. That's okay, John. We will close the session and you will have another chance to ask your question. But let's thank Doris once again for a beautiful talk and for answering all of these questions and really exciting work that you're doing. So thank you so much for spending your time this afternoon and morning with us.
Thank you so much, and it was wonderful to see you again, Dean.
Sure, absolutely. So if you can stick around afterwards for people who want to ask questions in private, but let me thank everybody who joined us this afternoon. If you want to ask questions in private, stick around. Otherwise, see you next time. Take care.